Have you ever noticed that some people seem to move through their later years with a kind of quiet ease, while others find themselves in a slow but steady decline that no single doctor's visit seems to fully explain? We tend to assume this is simply genetics or luck. But there is another possibility worth considering. What if the body is not gradually wearing out, but rather responding to conditions that either support or undermine its natural capacity for balance? What if many of the changes we attribute to aging are actually the result of accumulated imbalances that could have been prevented or even reversed? This is not a question of fighting against time, but of understanding something far more subtle: the body's own regulatory systems, which work constantly to maintain stability, and the factors that either help or hinder their function. Today, we are going to explore what happens when one of the most important of these regulatory systems begins to drift off course, and what the latest research suggests about restoring its natural balance. You are listening to the Longevity Library, where we archive the wisdom of healthy aging. The architecture of vitality. Understanding the body's innate capacity for balance. There is a question that becomes more meaningful with each passing decade. What allows some individuals to move through their later years with ease and resilience, while others experience a steady decline that seems to accelerate despite their best efforts? This is not merely a question of genetics or luck, though both play their part. It is a question about patterns of living, patterns of thinking, and most fundamentally, patterns of biology that either support or undermine the body's remarkable capacity for self-regulation. The human body is not a machine that inevitably wears out with use. This metaphor, though common, is deeply misleading. Machines degrade because they lack the ability to repair themselves. They cannot replace worn components, cannot adapt to changing conditions, cannot learn from experience and modify their function accordingly. The body does all of these things continuously, effortlessly, without conscious direction. Every moment of every day, countless processes work to maintain balance, repair damage, and optimize function. The question is not whether the body can maintain itself, but whether we are providing the conditions it needs to do so effectively. This distinction matters because it shifts the focus from fighting decline to supporting the systems that naturally work against it. Rather than asking what we can do to stop aging, a question that assumes aging is an enemy to be conquered, we might ask what we can do to create the conditions in which the body's own maintenance systems can function optimally. This is a more modest question, perhaps, but also a more productive one. It acknowledges that we are not in control of every biological process, but that we can influence the environment in which those processes occur. The language of balance. Every biological system operates within a range of optimal function. Body temperature, blood sugar, fluid balance, heart rate. Each of these fluctuates throughout the day, but remains within boundaries that support health. When any of these parameters moves too far from its optimal range, the body activates corrective mechanisms to restore balance. This capacity for maintaining internal stability despite external changes is called homeostasis, and it is one of the defining features of living organisms. The immune system, like every other system in the body, operates according to homeostatic principles. It must remain vigilant enough to detect and respond to threats, yet restrained enough to avoid attacking healthy tissues. It must ramp up quickly when danger appears, then return just as quickly to a resting state once the threat has passed. This balance between activation and restraint is maintained by specialized cells whose sole function is to apply the brakes when the immune response has run its course. These regulatory cells, known in scientific literature as T-regulatory cells, serve as the immune system's thermostat. They do not participate directly in fighting infections or eliminating abnormal cells. Instead, they monitor the intensity of immune responses and gradually dial them down when they are no longer needed. Without these cells, The immune system would continue mounting full-scale responses indefinitely, like a heating system that never shuts off once activated. When the regulatory thermostat fails, the immune system can begin targeting tissues that were never meant to be attacked. This is the fundamental pattern underlying conditions that involve the immune system turning against the body's own structures. The immune cells themselves are not inherently defective. They are simply responding to signals that tell them to remain active when they should be quieting down. The problem is not that the immune system is too strong, but that its off switch has stopped working properly. Understanding this distinction is important because it suggests a different approach to supporting immune health. Rather than trying to suppress immune activity broadly, an approach that inevitably affects protective responses along with harmful ones, we might focus on restoring the function of the regulatory cells that naturally maintain balance. This is not about weakening the immune system, but about helping it function as it was designed to function. The nutrient that speaks to genes. Among the many factors that influence regulatory cell function, one stands out for its far-reaching effects throughout the body. This nutrient, which is technically a hormone, participates in the regulation of thousands of genes and influences virtually every system in the body. Its receptors are present on nearly every cell type, and its effects extend from bone health to mood regulation to immune function. Yet despite its importance, a substantial portion of the population has difficulty maintaining adequate levels of this substance or, equally problematic, adequate sensitivity to its signals. The substance in question is vitamin D, though this name captures only a fraction of its biological significance. Unlike most vitamins, which function primarily as cofactors for specific enzymes, vitamin D acts more like a steroid hormone. It binds to receptors inside cells, travels to the nucleus, and directly influences which genes are expressed and at what levels. Through this mechanism, vitamin D helps determine how cells respond to their environment, including how immune cells interpret the signals that tell them whether to attack or stand down. For regulatory T cells to develop and function properly, they must receive adequate vitamin D signals. Without these signals, the cells that should apply brakes to the immune system fail to mature or, if they do mature, fail to function effectively. The immune system continues operating without its normal regulatory input, and the balance between activation and restraint shifts toward persistent, low-grade activity. Laboratory testing for vitamin D typically measures the circulating form of the nutrient, which reflects what is available in the bloodstream, but not necessarily what is happening inside cells. This distinction becomes important because cells can become resistant to vitamin D signals even when blood levels appear adequate. When vitamin D receptors become less sensitive, the nutrient cannot deliver its message effectively, and cells behave as though they are deficient even when blood tests suggest otherwise. This phenomenon of cellular resistance helps explain why some individuals continue to experience symptoms associated with low vitamin D function despite taking supplements and showing normal blood levels. The nutrient is present but cannot communicate with the cells that need its signals. Addressing this situation may require supporting the cellular machinery that responds to vitamin D, not simply increasing the amount circulating in the bloodstream. Inflammation itself appears to interfere with vitamin D sensitivity. When tissues are inflamed, the cellular response to vitamin D diminishes, creating a cycle in which inflammation reduces the signals that would normally help resolve it. This may be one reason why chronic inflammation tends to persist once established. The very mechanisms that should quiet it down have been disabled by the inflammatory state itself. The microbial dimension of immune education. The human body does not develop in isolation. From the moment of birth, and possibly before, we are colonized by microorganisms that will influence our biology in ways that researchers are only beginning to understand. The collective genome of these microbes, the microbiome, contains far more genes than the human genome, and these genes produce compounds that directly influence immune development and function. Among the most important contributions of the microbiome to immune health is its role in educating regulatory cells. Certain bacterial species appear to be particularly important for this function, producing metabolites that signal immune cells to develop regulatory rather than aggressive characteristics. These organisms effectively teach the immune system when to fight and when to stand down, providing ongoing instruction throughout life. Two bacterial species have attracted particular attention for their role in supporting immune regulation. The first, known scientifically as Bifidobacterium infantis, is one of the earliest colonizers of the human gut, typically acquired during breastfeeding. This organism helps establish the foundation for immune development and continues to influence immune function throughout life when it remains present in adequate numbers. However, this species is highly sensitive to disruption. Antibiotics, dietary changes, and other environmental factors can reduce its populations, and many adults show little to no colonization with this important organism. The second species, Lactobacillus reuteri, appears to have direct effects on the cells that regulate immune responses. This organism produces compounds that support the development and function of regulatory T cells while simultaneously suppressing the activity of more aggressive immune populations. Through this dual action, L. reuteri helps maintain the balance between immune activation and restraint that characterizes healthy function. Beyond its direct effects on immune cells, L. reuteri also influences the production of oxytocin, a hormone perhaps best known for its role in social bonding, but with broader effects on stress physiology. Oxytocin counteracts many of the effects of cortisol, the primary stress hormone, helping to maintain calm in the face of challenges. This matters for immune health because cortisol, when chronically elevated, suppresses immune function broadly, affecting both protective and regulatory responses. By supporting oxytocin production, L. reuteri may help maintain appropriate immune function even during periods of stress. The loss of these beneficial organisms from the modern gut microbiome represents a significant change in human biology. Estimates suggest that a large majority of the population lacks meaningful colonization with B. infantis, and the proportion missing L. reuteri may be even higher. This widespread absence of organisms that have co-evolved with humans throughout our evolutionary history may have consequences for immune regulation that we are only beginning to appreciate. Restoring these organisms to the gut is not simply a matter of consuming them in supplement form. The survival and establishment of beneficial bacteria depends on providing them with appropriate conditions, the right nutrients, the right environment, and protection from factors that would eliminate them. Fermentation of dairy products, a traditional food preparation method used by cultures around the world, provides one means of supporting these organisms and enhancing their ability to colonize the digestive tract. The mineral that modulates immune balance. Trace minerals receive less attention than vitamins in discussions of health, yet they play equally critical roles in biological function. These elements are required in such small amounts that deficiency may take years to develop. But when deficiency occurs, the consequences can be profound. Among the trace minerals most relevant to immune regulation is selenium, an element whose name derives from the Greek word for moon. Selenium's role in immune function centers on its incorporation into proteins that regulate oxidative stress and immune activity. These selenoproteins, as they are called, help protect cells from the damage caused by normal metabolic processes and immune responses. They also directly influence the behavior of immune cells, helping to determine whether responses will be aggressive or restrained. When selenium is inadequate, the balance between different immune cell populations shifts. The cells that drive inflammation and tissue damage become more numerous and more active, while the cells that regulate immune responses become less effective. This shift mirrors the pattern seen in conditions where the immune system targets the body's own tissues, suggesting that adequate selenium may be particularly important for maintaining appropriate immune restraint. Selenium also influences the production of autoantibodies, the antibodies that mistakenly recognize the body's own structures as foreign. These self-reactive antibodies contribute to tissue damage in many conditions involving immune system overactivity. Adequate selenium levels are associated with lower production of these antibodies, potentially reducing the burden on tissues that might otherwise be targeted for attack. The amounts of selenium required for these effects are modest, measured in micrograms rather than milligrams. A single Brazil nut, grown in soil with adequate selenium content, can provide a day's worth of this mineral. Seafood, organ meats, and eggs also contain selenium, though the amount varies depending on the selenium content of the soil where animals were raised or where their feed was grown. This geographic variation means that individuals living in regions with selenium-poor soil may not obtain adequate amounts from locally grown plant foods. As with many nutrients, there is a range of intake that supports optimal function, and both deficiency and excess can cause problems. Selenium toxicity, though rare, can occur with excessive supplementation and produces its own set of adverse effects. The goal is not to consume as much as possible, but to ensure adequate intake within the range that supports healthy function. The reset that occurs in absence. Perhaps the most powerful intervention for supporting immune regulation involves not adding something to the body, but temporarily removing something. When food intake ceases for an extended period, the body initiates a series of changes that fundamentally alter its function. These changes include the activation of cellular cleanup processes, the reduction of inflammation, and eventually, the regeneration of new cells to replace those that have been damaged or have simply reached the end of their useful life. The process of cellular cleanup, known scientifically as autophagy, involves the breakdown and recycling of damaged proteins and organelles within cells. This housekeeping function operates at a baseline level continuously, but becomes more active during periods of nutrient scarcity. When the body senses that food is not available, it begins clearing out cellular components that are no longer functioning optimally, using their components to fuel essential processes and building new structures from the recycled materials. For immune cells, this cleanup process has particular significance. Old or dysfunctional immune cells, including those that may have been programmed toward inappropriate responses, are targeted for removal. Their components are broken down and recycled, reducing the population of cells that might contribute to immune imbalance. This creates space for the generation of new immune cells, produced from stem cells that become more active during the recovery period following a fast. The generation of new immune cells following a fast differs from the normal process of immune cell production in important ways. The new cells arise in an environment that has been cleared of much of the inflammatory signaling that was present before the fast. They develop without the same exposure to factors that might program them toward aggressive responses, and they may therefore be more responsive to regulatory signals that promote restraint and balance. The duration of fasting required to trigger these regenerative effects appears to be longer than the intermittent fasting periods that have become popular in recent years. While shorter fasts provide benefits for metabolic health and may reduce inflammation to some degree, the more profound effects on immune cell populations typically require periods of 3 days or longer without caloric intake. During this time, the body progressively shifts its metabolism, first using stored carbohydrates, then shifting to fat breakdown and ketone production, and finally entering a state where cellular cleanup and regeneration become prominent. This is not a practice to be undertaken lightly or without appropriate preparation and supervision. Prolonged fasting affects fluid and electrolyte balance, can alter the effects of medications, and may not be appropriate for individuals with certain health conditions, for those who can safely undertake it under proper guidance. However, periodic prolonged fasting offers a means of resetting immune function that cannot be achieved through any other intervention. The integration of influences, the factors that support immune regulation, do not operate in isolation. They interact with each other, amplifying beneficial effects when present together and potentially creating gaps when any are missing. Understanding these interactions helps explain why single interventions often produce disappointing results and why a more comprehensive approach may be necessary for meaningful change. Vitamin D provides the foundational signal that allows regulatory cells to develop and function. Without adequate vitamin D signaling, these cells cannot mature properly, and other interventions have limited effect. The regulatory cells simply are not present in sufficient numbers or with sufficient function to respond to other supportive signals. The gut microbiome, particularly those species that have co-evolved with humans throughout our history, provides ongoing instruction to immune cells. These organisms produce metabolites that directly influence regulatory cell development and function, effectively teaching the immune system when to activate and when to restrain itself. When these organisms are absent, immune cells lose access to this ongoing education. Selenium supports the enzymatic systems that protect cells from oxidative damage and directly modulates the balance between different immune populations. Adequate selenium helps ensure that regulatory cells can function in environments where they might otherwise be overwhelmed by oxidative stress or inflammatory signals. Prolonged fasting creates opportunities for renewal that cannot be achieved through nutritional interventions alone. By removing old, dysfunctional cells and stimulating the production of new ones, fasting resets the immune system in a way that allows other interventions to work more effectively. The new cells that emerge following a fast are more responsive to regulatory signals and less likely to have been programmed toward inappropriate responses. The order in which these interventions are introduced may matter. Establishing adequate vitamin D signaling before attempting prolonged fasting ensures that the regulatory cells produced during the regeneration phase will have the signals they need to mature properly. Supporting the gut microbiome before and after fasting helps ensure that beneficial organisms will colonize the newly cleared environment. Addressing selenium status ensures that the enzymatic systems protecting new cells will function optimally. The pattern of healthy function. When these various influences align, the immune system operates differently. It responds appropriately to genuine threats, mounting effective responses that eliminate pathogens without causing excessive collateral damage. When the threat passes, the response subsides, returning to a baseline state of quiet vigilance. The system remains ready to respond, but does not remain constantly active. This pattern of appropriate response followed by return to baseline characterizes healthy function in all biological systems. The cardiovascular system responds to increased demand by raising heart rate and blood pressure, then returns to resting levels when demand decreases. The digestive system ramps up activity during meals, then quiets down between meals. The nervous system activates during challenges, then restores calm when challenges pass. In each case, the ability to both activate and deactivate appropriately is essential for long-term health. Immune function follows the same principle. The problem in conditions where the immune system targets the body's own tissues is not that the immune system activates. Activation is necessary for protection, but that it fails to deactivate appropriately. The off switch has stopped working, and the system remains active when it should have returned to rest. Supporting the off switch, rather than trying to suppress the entire system. represents a fundamentally different approach to maintaining immune health. This approach acknowledges that the immune system's basic design is sound and that the goal is to restore its normal regulatory function, not to override it with powerful suppressive agents. It is a more subtle approach, perhaps, but one that works with the body's natural design rather than against it. The question of individual variation. Not everyone responds to these interventions in the same way, and understanding why reveals important principles about biological individuality. Genetic variations influence how efficiently vitamin D is activated and how effectively cells respond to its signals. Differences in the gut microbiome, shaped by early life experiences, diet, and medication history, affect which bacterial species are present and how they interact with the immune system. Variations in selenium metabolism influence how much of this mineral is needed to support optimal function. These individual differences mean that there is no single protocol that works equally well for everyone. What restores balance in one person may be insufficient for another, and what works at one stage of life may need adjustment as circumstances change. The goal is not to find a universal formula, but to understand the principles well enough to apply them appropriately to individual situations. This understanding requires paying attention to how the body responds to different interventions and adjusting accordingly. It requires patience, because biological changes take time and because the effects of supporting regulatory function may be subtle at first. It requires a willingness to learn from experience, to notice what shifts and what remains unchanged, and to modify approaches based on observation rather than expectation. The context of daily life. The factors that support immune regulation do not exist in a vacuum. They are embedded in the larger context of daily life, where sleep, movement, social connection, and meaning all influence biological function. Understanding this context helps explain why focusing on isolated interventions often produces disappointing results and why a more comprehensive approach may be necessary. Sleep, for example, is when much of the body's repair and maintenance work occurs. During deep sleep, the brain clears metabolic waste products that accumulate during waking hours. Hormones that support tissue repair are released. The immune system processes information about threats encountered during the day and adjusts its responses accordingly. Without adequate sleep, these processes are impaired, and the benefits of other interventions may be diminished. Movement, too, influences immune function in ways that extend beyond the obvious benefits for cardiovascular and musculoskeletal health. Regular physical activity appears to support regulatory immune function, perhaps through its effects on inflammation and stress hormones. Sedentary patterns, by contrast, are associated with persistent low-grade inflammation that may interfere with immune regulation. Social connection, long recognized as important for mental health, also influences physical health through mechanisms that are only beginning to be understood. Loneliness and social isolation are associated with increased inflammation and impaired immune function, effects that may be mediated through stress hormone pathways. Meaningful social engagement, by contrast, appears to support healthy immune regulation. The sense of purpose and meaning, the feeling that one's life has direction and significance, has been linked to better health outcomes across multiple studies. This association may operate through multiple pathways, including effects on stress physiology, health behaviors, and the ability to navigate life's challenges without becoming overwhelmed. Whatever the mechanisms, the evidence suggests that a sense of purpose matters for physical as well as mental health. The time factor. Biological change takes time. Cells turn over on schedules that range from days for some immune cells to years for others. Tissues remodel slowly, responding to altered conditions over weeks and months. The effects of supporting regulatory function may not be immediately apparent, and impatience can lead to the abandonment of approaches that would eventually produce meaningful change. This is particularly true for interventions that work by supporting the body's own regulatory systems rather than by directly suppressing unwanted activity. Suppressive approaches often produce rapid effects because they override natural processes. Supportive approaches work more slowly because they must work through those same processes, gradually shifting the conditions that determine how the system operates. The timeline for meaningful change depends on multiple factors, including the starting point, the consistency with which supportive practices are maintained, and the presence of other factors that either support or undermine regulatory function. For some individuals, shifts may be noticeable within weeks. For others, months may be required before changes become apparent. In either case, patience and consistency are essential. The question of maintenance. Once regulatory function has been restored, maintaining it requires ongoing attention to the factors that support it. Vitamin D status must be maintained through adequate sun exposure or supplementation. The gut microbiome must be supported through diet and, when necessary, through reintroduction of beneficial organisms. Selenium intake must remain adequate to support the enzymatic systems that protect cells and regulate immune responses. Periodic fasting may be needed to reset the system when stresses accumulate. This ongoing attention is not a burden, but simply a recognition that health is not a static achievement. It is a dynamic process that requires ongoing engagement, much like maintaining a garden or tending a fire. The conditions that support healthy function must be maintained, or function will gradually drift toward less optimal states. The good news is that these maintenance activities, once established as habits, require less conscious effort over time. The body adapts to regular patterns, and what initially required deliberate attention becomes increasingly automatic. The microbiome stabilizes in response to consistent dietary patterns. Vitamin D status, once optimized, can be maintained with less frequent attention. The skills developed during prolonged fasts can be applied more easily in the future. The limits of knowledge. It is important to acknowledge what is not known as well as what is known. The science of immune regulation is still developing, and many questions remain unanswered. The interactions between different factors are complex and not fully understood. Individual variation means that what works for one person may not work for another, and predicting who will respond to which interventions remains challenging. This uncertainty is not a reason for inaction, but a reason for humility. It suggests that approaches should be undertaken with an attitude of experimentation, paying attention to results and adjusting accordingly. It suggests that dogmatic adherence to any single protocol is unlikely to serve well, and that flexibility and observation are essential companions to any health practice. It also suggests that the most reliable guide may be the body itself, the sensations of well-being, the patterns of energy and fatigue, the quality of sleep, the ease of movement. These subjective experiences provide information that no laboratory test can capture. Learning to attend to this information, to notice what shifts in response to different practices, is itself a skill that can be developed and refined over time. The larger perspective. Throughout human history, people have maintained health without access to modern supplements, laboratory testing, or detailed scientific understanding of immune function. They did so by living in ways that naturally supported the body's regulatory systems, eating foods that provided necessary nutrients, maintaining activity patterns that kept tissues resilient, experiencing the cycles of feasting and fasting that reset biological processes, and remaining connected to communities that provided meaning and support. Modern life has disrupted many of these patterns while providing others. We have access to information and resources that previous generations could not imagine. We also face challenges they did not: chronic stress, sedentary occupation, processed foods, social isolation that may undermine the very systems we are trying to support. The task, then, is not to reject modernity, but to learn from tradition while using the tools that modern knowledge provides. It is to understand the principles that have always supported human health and to apply them in circumstances that may be very different from those in which they originally operated. It is to recognize that the body's capacity for self-regulation is remarkable but not unlimited, and that creating the conditions for that capacity to express itself fully is perhaps the most important thing we can do for our long-term health. This perspective shifts the focus from fighting against aging to supporting the systems that naturally work toward balance. It acknowledges that decline is not inevitable, that many of the changes commonly attributed to aging are actually the result of accumulated imbalances that could have been prevented or reversed. It suggests that the goal is not to live forever, but to live well, maintaining function and vitality throughout the years we are given. The integration of knowledge. The factors discussed here: vitamin D signaling, the gut microbiome, selenium status, and periodic fasting represent different aspects of a larger picture. They are not isolated interventions, but interconnected elements of a system that supports immune regulation. Understanding how they fit together provides a framework for thinking about health that is more useful than any single intervention could be. Vitamin D provides the signal that regulatory cells need to develop and function. Without this signal, the other factors have limited effect because the cells that should respond to them are not present in adequate numbers or with adequate function. The gut microbiome provides ongoing education to immune cells, producing compounds that directly influence regulatory function. These organisms have co-evolved with humans throughout our history, and their absence may represent a significant change in the conditions under which human immune systems developed. Selenium supports the enzymatic systems that protect cells from oxidative damage and directly modulates the balance between different immune populations. Adequate selenium ensures that regulatory cells can function effectively even in challenging environments. Prolonged fasting provides a periodic reset, clearing out old or dysfunctional cells and stimulating the production of new ones. This creates opportunities for the other interventions to work more effectively, as new cells are more responsive to regulatory signals. Together, these factors create conditions in which the immune system's natural regulatory mechanisms can function optimally. They do not override the system, but support it, working with its design rather than against it. The path forward. For those who find this framework resonant, the question becomes how to apply it in daily life. This is not a question with a single answer, because circumstances differ and because the body's responses are individual. But some general principles may serve as guideposts. Begin with the factors that provide foundation. Ensuring adequate vitamin D signaling, whether through sun exposure or supplementation, creates the conditions under which regulatory cells can develop and function. This may require higher intake than standard recommendations suggest, and it may require attention to factors that influence cellular sensitivity as well as blood levels. Consider the state of the gut microbiome. Are the organisms that have historically supported immune regulation present in adequate numbers? If not, what might be done to reintroduce them and to create conditions that allow them to thrive? This may involve dietary changes, targeted use of fermented foods, and protection from factors that would eliminate beneficial organisms. Address nutritional foundations, including trace minerals like selenium that support the enzymatic systems protecting cells and regulating immune responses. This does not require elaborate supplementation, but does require attention to whether dietary intake is adequate given individual circumstances and the selenium content of locally grown foods. Consider the potential role of periodic fasting as a means of resetting immune function, for those who can safely undertake it under appropriate guidance. Prolonged fasting offers effects that cannot be achieved through other means. For others, modified approaches may provide some benefits with less risk. Throughout this process, pay attention to the body's responses. Notice what shifts, what remains unchanged, what improves, and what does not. Use this information to adjust approaches, recognizing that the goal is not adherence to a protocol, but support of the body's own regulatory capacity. The calm of understanding. There is a certain peace that comes from understanding how the body works. The confusion and frustration of unexplained symptoms. The sense that health is slipping away despite best efforts. The feeling of being at war with one's own biology. These experiences can be profoundly unsettling. Understanding that the body is not an enemy, but a system seeking balance. That symptoms are not attacks, but signals. That many of the changes we fear are not inevitable, but reflect accumulated imbalances that can be addressed. This understanding changes the relationship with one's own body. It replaces fear with curiosity, frustration with patience, helplessness with engaged attention. It acknowledges that we are not in control of every biological process, but that we can influence the conditions in which those processes occur. It recognizes that the body's capacity for self-regulation is remarkable, but not unlimited, and that our role is to provide the conditions that allow that capacity to express itself fully. This is not a promise of perfect health or unlimited years. It is something more valuable, a framework for understanding that makes sense of experience and provides guidance for action. It is a way of thinking about health that is coherent, grounded in observation and reason, and respectful of the body's complexity. And it is a reminder that the body, given half a chance, will work toward balance. Our task is to give it that chance. The central insight, the one worth carrying forward, is this: the body is not your enemy, and it is not simply wearing out. It is a system seeking balance, and given the right conditions, it will work toward that balance with remarkable persistence. The questions we have explored today are not about quick fixes or dramatic interventions, but about creating the environment in which the body's own regulatory capacity can function as it was designed to function. If this conversation has sparked something in you, a curiosity, a recognition, a question of your own, we would welcome your thoughts in the comments. And if the idea of building a personal library of health wisdom appeals to you, we invite you to explore the other recordings here and to subscribe so that new editions find their way to you. This is the Longevity Library. Listen, learn, and live longer.